Good morning, everyone. It gives us immense pleasure to announce that the Department of Microbiology, Tripura University, is organizing an online lecture series on different aspects of life sciences as part of the celebration of Azadi Ka Amrit Mahotsav. We would have eminent speakers from around the globe who'd be talking about the latest development in life sciences in their respective field. We, the members of the department, which includes our students, our scholars, our staff, and our faculty member, welcome you all to this online webinar series. As the first speaker today, we have with us Dr. Radhashree Maitra from Yeshiva University, New York. And she's going to talk about Reovirus as a therapy for colorectal cancer, role of autophagy. Welcome, Dr. Moitro. About Dr. Moitro, our first speaker. Dr. Moitro got her bachelor's in chemistry and master's in biochemistry with a specialization in molecular biology from Calcutta University. She did her PhD in biophysics, molecular biology, and genetics from Calcutta University in 1997 with funding from UGC through the UGC Net Award, then CSIR through the SRF CSR India in 1989 and 1993, respectively. She did her postdoctoral work at Washington University in St. Louis and then at Albert Einstein College of Medicine of Yeshiva University, New York. Dr. Moitro has 43 original peer-reviewed publications. She's currently an associate professor of biology at Yeshiva University and a senior scientist at Montefiore Medical Center. Her current research is geared towards elucidating the molecular basis of the therapeutic efficacy of unique reovirus, the double-stranded RNA virus in KRAS mutated colorectal cancer. Dr. Moitro has been associated with our department for quite a few years now, where she was the visiting scientist who had been here with the ASM Visiting Scientist Award for teaching our students and the scholars about the recent advanced techniques that are used in microbiology a couple of years back. And this support or hand-holding continues. Thank you, ma'am, for accepting this invitation. We understand it's midnight at your place, and yet you have taken this trouble of being our first speaker. So, welcome, ma'am.
Thank you, Dr. Roy Chaudhuri. It's a pleasure to be here. A very good morning to my viewers, students, friends, colleagues. It's 11 a.m. there. It's 1 a.m. 25th September here. So we are on the same date, although we are on the two opposite sides of the planet. I would first like to thank the organizers of this meeting for providing me this opportunity to present my work. And special thanks goes to Dr. Roy Chaudhuri because she is the pioneer in inviting me and keeping me attached to the university. So we were trying back and forth to make the slideshow. It looks like the slideshow is a little bit difficult, but that should not affect us in any way in making our presentation. And I think everyone should be able to understand my presentation. There are a few videos. If we get a chance, Dr. Roy Chaudhuri is going to show them from the other end. And here I start. The title of my today's presentation is Reovirus as a therapy for colorectal cancer and the role of autophagy. Lately, I have been working with autophagy. The photographs that you see on the screens, these were taken by me back in 2010, 2011. Since then, I have made we have many good pictures and data, but somehow these two are very close to my heart. And whenever I get an opportunity, I try to show them. This is Reovirus by transmission electron microscopy, and on the right you see the reovirus by scanning electron microscopy, where the virus is budding out of the cells. So moving to the main topic, I work with colorectal cancer, and just to take a quick look at cancer burden worldwide, we know we are nowadays almost the entire population is aware of the disease, the consequences, and what we can do, and how to avoid and how to manage the disease. So colorectal cancer is the third most the cancer that is seen. It is most commonly diagnosed and leading cause of cancer related death worldwide. It is expected that by end of 2030 there'll be a 60% more increase with 2.2 million new cases, and the situation here is quite grim. So the genetically modified food, the comfort food that we eat, the fertilizers, the preservatives, and all that we consume knowingly, unknowingly, are really having serious consequences on the epithelium of our colon, and awareness is absolutely necessary. So here we see the Global Cancer Observatory data, which shows that the estimated new cases and the estimated number of deaths, and this number is quite grim. It consists of almost 10% of all the types of cancer and happens next to lung. So looking at the cancer statistics in America, a first world country where the incidence is very high, we would not look at the huge numbers over here, but let's look at the daily rate. The daily rate of 5,000, approximately 5,000 new cases with more than 1,500 people dying daily only in America. The situation is quite serious. So what are the treatment options? So colorectal cancer, so initially, some 70, 80 years back, there were higher incidence of stomach cancer because people used to preserve food by using pickles and preserving them in all different old strategies. But as the refrigerators came in, stomach cancer significantly decreased and colon cancer started to emerge. The reason behind it is that now we were preserving food for longer time and consuming it at a date which is, so the expiry dates were extended and we were consuming it much later than when it was prepared, and that was one of the reasons why the cases of colorectal cancer incidence were much higher. So if we look at the CRC, colorectal cancer, CRC is the abbreviation I'm going to use throughout. So about 55% of the CRC patients are KRAS wild type and 45% are KRAS mutant. Those which are KRAS wild type, they depending on the stage of diagnosis, they are given the frontline treatment. After the frontline treatment, they are given the biologics, the monoclonal antibodies. The approval of monoclonal antibodies for treatment of KRAS wild type CRC cancer had significant improvement in the survival, and the median survival increased from an average of 12 months to 19 months. So it was a significant 7-8 months of survival improvement. On the other hand, those that had KRAS mutations were not so fortunate. So after the standard frontline treatment, the survival was significantly reduced with the monoclonal antibodies, with the biologics. So although people were happy because the biologics have much less side effect than the chemical chemotherapies, the use of monoclonal antibody was not that effective in KRAS mutant cancers. Not only that, they were actually detrimental. So this was the first test which the United States FDA, the Food and Drug Administration, made it mandatory that before the monoclonal antibody is administered, every CRC patient has to be tested for the KRAS status. If it's a mutant KRAS, then they were not allowed to be given the monoclonal antibodies. Now, unfortunately, this cohort of patient does not have any other approved therapy to be given, to be administered when the frontline treatment fails. So essentially, not only because of the oncogenic KRAS mutation, the cancer is aggressive in nature, but it also there were no treatment options available for this cohort of patient. And this is the basis of my research, where I want to address this issue of patients having CRC with KRAS mutation and not having being able to have a treatment option as opposed to the KRAS wild type. So you see, this is my research field. So KRAS, let's talk a little bit about KRAS. Why does KRAS make the cancer so aggressive, and why is it not why can it not be handled? So it's a 21 kilodalton protein with multiple hotspots. Of course, there are different mutants, and all these mutant forms cause aggressive form of CRC. For last decade, scientists have tried many ways to harness this target, but the shape of KRAS is such that it is almost non-druggable. Not only that, each of the mutants are different in their conformation, and hence a single drug is not viable for all the mutations. Lately, I think three or 4 months back, one of the small molecules have been approved in the clinic for only one type of mutation, G12C, but the incidence of G12C mutations is much low in CRC. So the CRC patients are still at the same point at which they were even before the small molecules were approved. Mutations in KRAS being an oncogene, a single, so even if it's heterozygous, that is lethal. So mutations are embryonically lethal, and hence we did not have good preclinical animal models where different drugs can be tried and verified before it moves to the clinic. All the same, the crystal structure of KRAS has been solved, and we know KRAS from as a protein. We know the structural details, and we performed all different simulations, which I'll show you in a minute. So here we see a pie chart which shows all the different KRAS mutations that are often found in patients. So there are many different types of mutations, and there are a few hotspots like codon 12 and 13 are very prominent and often gets mutated. What happens when KRAS is mutated? EGFR, so monoclonal antibody, the cetuximab. Cetuximab binds to acts as a ligand to the epidermal growth factor receptor, EGFR. KRAS is downstream of it. It's a small molecule GTPase, which is bound to the inside of the membrane, and it sends out signals through a cascade and turns on proliferation and growth. So ideally, if anti-EGFR antibodies are used, this whole signaling of growth should be stopped, and KRAS should not be able to convey any signals to the nucleus. That is what happens when KRAS is not mutated. But when KRAS is mutated, it is extremely proactive, and if if one of the pathways is inhibited by inhibitors like MEK inhibitor or a ERK inhibitor, it takes up another pathway, the PI3 kinase pathway, and makes it even more aggressive. So the use of anti-EGFR antibody or the biologics, which were extremely beneficial for KRAS wild type CRC patients, were actually very detrimental. So if this pathway was blocked, another pathway was activated, and that led to even worse consequences. So this I had as a animation, but unfortunately I'm unable to show the animations. So this is the single amino acid sequence. We have the codon 12 and 13. Codon 12 has a glycine. It's a small amino acid which is hydrophobic. When it is converted to aspartic acid, then what happens is that the entire conformation of the protein changes. So essentially, it's a KRAS is a GTPase. It binds to the GTP, it breaks the gamma phosphate bond, and in the process sends out the signal. Once GDP is formed, the signaling is stopped. In the mutant proteins, the GTP hydrolysis is prevented. And then there's a constant signal going on because the GTP is bound to KRAS, and that binding, as long as it's bound and not hydrolyzed, it is constantly sending out the signals, which aggravates the growth and the proliferation. So we did, so this was the study. This is my latest study. This I did during the pandemic. We were not allowed to go to the lab. New York was epicenter for quite a few months, and everybody was terrified with the pandemic. So that was when we studied it. We used a very fancy platform, the Schrodinger platform, and we did molecular dynamics simulation, trying to understand what exactly is happening with the protein and its conformation. So we selected 7 mutants at codon 12 and 13, and when we performed the simulation and we used GTP as a ligand, we found that GTP does not bind into the pocket of the KRAS protein. So this is G12C mutant. And the protein conformation around the pocket has also changed. And in this entire process, we see over here is the wild type. I'm sorry if it is not so clearly visible. So what do we see here? In the wild type, the GTP actually binds to the pocket and gets into a conformation where it can be hydrolyzed, and that is not happening in any other mutants, and hence they are so aggressive. So this is the ribbon form of the molecule, and actually let me make a quick try and see if I do a slideshow here. Are you able to see okay, this is where we are having the structural simulation. I'm not sure if my viewers can see the animation. Can you see, Shaun? So with the simulation, we could see exactly where the ligand is binding and how.
The animation is not visible because it's not in slideshow.
All right. So this is, I move on. And this is where we see the G13D mutation, which is also a very common mutation. And we see the wild type molecule. So even without the animation, let's see. Is this happening, Shaun?
Yes, it is.
Okay, I'm glad that at least. So here we have on the left, we have the G13D mutation, and on the right is the wild type. So what do we see? If you look at it carefully, the blue is the ligand, the GTP molecule. It is far away from the pocket where it should bind. Whereas in the wild type, you see the ligand is sitting right at the pocket where it should bind, and that binding in that particular conformation is essential for the GTP to get hydrolyzed. Once GTP is hydrolyzed and the event is completed, the signaling stops, and then until and unless the next GTP molecule comes, the signal is not initiated. So the next, I'm sorry, just a second. Let's see if so here we look at the ligand now. Here we look at the ligand, which is the GTP molecule, and we are trying to see the amino acid contact. So KRAS protein, which are the amino acids which are in contact with GTP, and what do we see? So first I'm going to point out to the wild type molecule. The wild type molecule is on the extreme right hand corner. If you would follow my cursor, you can see it. You would see that the gamma phosphate is totally free. There are no amino acids which are tightly binding to it, and it is almost contact free. Whereas in most of the other mutants, what we see is that the gamma phosphate is at a contact of above 80%. So it's tightly bound. The gamma phosphate in most cases is tightly bound, and that prevents the hydrolysis. So these are at position 12. We also have G13D mutation where we don't see the gamma phosphate being so much occluded by the amino acids of KRAS, but this particular conformation and also the charge distribution, if you would see that the colors at the bottom shows the charges. So G13D mutation, the entire molecule has a positive charge, and that is the one that prevents it from further hydrolysis. So we looked at the ligand, we looked at KRAS protein, we did the simulation. This was a research which I probably would not have done if there was no pandemic, but it did give us a great insight into KRAS, and I'm sure this is going to help us in designing molecules, small molecules, which will be very specific to binding to this newly formed hydrophobic pocket or the GTP binding pocket. So we have already started looking at those molecules. So here is another piece of data. Here we do the root mean square deviation to measure the average change of displacement of a selection of atoms for a particular frame with respect to reference frame. So essentially here what we are measuring, the blue one, the blue or the green graph lines that you are seeing is for GTP, and the red is KRAS. And for wild type, we see that the binding constant is just less than 5 nanoseconds. So the GTP binds for a few seconds, gets hydrolyzed, and it's easily released. Whereas in the other mutants, we see longer and tighter binding, like G12A, G12S, these are all, or G12V, look at it. It's constant, it's bound, and it is bound, and the protein ligand contact is for quite a long time. So with this we understand that while wild type display no stability, the mutants are quite stable with their ligands. We further looked into it, and we looked at the residue 12. Residue 12 is glycine again, and the angle is quite critical. So while KRAS is studied, it is studied for 3 regions: the switch 1, which is between 30 and 38 amino acid, switch 2, which is between 59 and 67 amino acids, and also the angle of the glycine. We see that at wild type, the glycine has a much higher angle. The next one is G12C, which is 115.7. For wild type, it's 134.4. This is quite a significant observation. The only small molecule which was approved for clinical trial few months back targets G12C. G12C actually is a mutation that gives a conformation which is more close to the wild type than rest of the mutations. So each of these molecules have different angles, and they are all different from wild type, which is at an angle of 134.4. So this angle allows the gamma phosphate to be available for hydrolysis. So, with this, although my topic says that it's reovirus and autophagy, why have I been talking about KRAS? Because reovirus preferentially works and performs oncolysis in KRAS mutated CRC. So all these past 10 years I have been studying reovirus, but and I have known that it is mostly active in KRAS mutated, but now I know the structure, the molecular dynamics of KRAS in greater details, and the next simulation I'm going to run is with the reovirus molecule, just to see if reovirus directly affects KRAS binding and binding of KRAS to GTP. So a little bit about reovirus. Reovirus is a double-stranded RNA virus. This virus has segmented genome. There are 10 different segments of the genome, and it's naturally oncolytic virus. This virus does not go, it is not a retrovirus. It does not go through DNA to RNA and RNA to RNA to DNA and DNA to RNA again for transcription translation, which retroviruses like HIV does. So this is a double-stranded RNA genome. One of the strands is the positive strand that codes for the protein, and different segments are functional at different points, which makes its biology, makes its genetics a little bit complicated, but also interesting at the same time. It's a naturally occurring oncolytic double-stranded RNA virus. It's not transgenic or recombinant, not man-made. It's very, very mildly pathogenic. This is an added advantage for the clinicians and for us to try it out, because cancer patients are usually sick and we don't want to administer any biologics which can give other side effects. So most of us who have been in the third world country have been exposed to reovirus, and we all have antibodies against it. So it's more potent in transformed cells, including the oncogenic KRAS mutation, and development was a good choice, understanding that they were not KRAS mutated CRC patients were not allowed to have the regular biologics like anti-EGFR antibody, cetuximab, and panitumumab. Developing another biologic made sense, and this was a very good platform to study both the virus and the etiology of the cancer. So this was the current working model. So it still holds that way. We published it in 2011. So essentially, if reovirus enters cells by phagocytosis, enters the endosome, it gets the capsid protein, outer coat protein is removed, and the double-stranded RNA is released. From there, the coding strand, the transcript is synthesized, and then if it's a KRAS wild type situation, if the CRC cell is KRAS wild type, the translation is blocked, and the translation is blocked with the help of a transcription factor which gets phosphorylated. We have the protein kinase receptor that also gets phosphorylated, and this entire pathway prevents the translation of the transcript. Whereas when it's a KRAS mutant, then PKR is not phosphorylated, PKR becomes ineffective, and translation continues and viral assembly takes place. And finally, when enough viruses have accumulated, the cell releases the infective virions, and in the process, it kills the cells. So when reovirus administered, it only goes and causes enters and causes oncolysis to the transformed cells which are cancerous cells which have KRAS mutation. The normal cells are usually spared. It does not mean that the normal cells are not infected. The normal cells, as I show you in this cartoon, gets infected, but then translation and formation of active virions is not allowed by the host. So so what would be a good strategy for therapeutic development? This slide was also made quite a few years back when we were discussing how to improve reovirus and how to make it useful for KRAS mutated CRC patients. So one way to study was to use it in combination with chemotherapy and radiation therapy in cell lines and preclinical models. And we already published the data of its synergy with irinotecan in patients and cell lines back in 2014, if I'm not wrong. So the preclinical data was published then. The clinical phase 1 trial was published last year, and I'm sorry, it showed reovirus showed quite good efficacy when used in combination. The other strategy could be how to harness or how to control the cellular processes towards improving viral efficacy. So by improving infectivity, by allowing better propagation. So one of the strategies at that point that we took at that point was the regulation of host TLR3. So toll-like receptors are innate immune molecules that senses viruses, especially double-stranded RNA viruses are sensed by TLR3, and TLR3 is found on the endosomes. So if downregulation of TLR3 is not usually detrimental for the cells, so TLR3 mutation does not kill a cell or a human cell, but if TLR3 is downregulated, then that allows better infectivity of TLR3, and we have already shown that, and that data was published, I think, in 2016. The other way to study better is to develop a genetic mouse model, which actually recapitulates the human colon carcinoma with KRAS mutation. So all these years, we did not have a robust genetic mouse model where we could do preclinical studies. Mice with KRAS mutations often had embryonically lethal situations. So there were no pups which were born with KRAS mutation and CRC. That also was being tried for 10 years and was not successful. Finally, the third strategy is to manipulate the virus by a process called reverse genetics. A lot of genetics have been done with DNA viruses. Viruses are double-edged swords. So here I'm talking about oncolytic viruses, but viruses we know can also cause cancer. HPV, human papilloma virus, Epstein-Barr virus, they are known to be quite potent in causing cancer. But so viruses are double-edged sword. They can create cancer, they can cure cancer. So how better to handle it? DNA viruses, as I just mentioned, has been engineered a lot, both as a vehicle for drug deliveries and like adenovirus, and also for oncolytic properties. RNA viruses, double-stranded RNA viruses, especially with segmented genomes, are a little bit harder, but still it is something we can always try to address. So this was where we published in 2014, where we showed that KRAS mutant cells are much more effective with reovirus, so they are better killed, and with a significant cell killing when the cells are KRAS mutant. We used a panel of 13 cell lines of KRAS mutation and about 8 cell lines CRC cell lines which were KRAS wild type, and we found a much better cell killing with reovirus. Reovirus was generally used between 2 to 5 MOI. MOI means multiplicity of infection. And we also showed that not only did reovirus function as a single agent, it also nicely was synergistic with other chemotherapy like irinotecan. So the combination is synergistic, which essentially means that it shows an effect which is more than just simple additive effect of the two agents. So whatever effect irinotecan caused and whatever effect reovirus caused, the combination had a much better efficacy than that. And we showed that in KRAS wild type, it was not that significant, whereas in KRAS mutant, the synergy was quite significant. So we started with apoptosis. We showed that reovirus causes apoptosis, and that was how we were proceeding with our research. So we, at that point, we also started looking at the electron microscopy of reovirus. That was something which was very exciting. I came back to the same slide which I discussed as I started my presentation. So we looked at different cell lines which were infected with reovirus, both by transmission electron micrograph as well as scanning electron micrographs. So we did see these beautiful structures. These are called the viral arrays. We saw the viral arrays at around 30,000 magnification, and this is around 3,000 magnification for scanning electron microscopy. This was very impressive. We were very, very happy to see it, actually thrilled at that point to see it. When we saw the viral arrays, we saw that the viral arrays were seen only in the KRAS mostly seen significantly higher in KRAS mutant cells as compared to KRAS wild type. So we used isogenic CRC cell lines. So HCT116 and HKE3, they are isogenic, meaning they are identical to one another, except that one had KRAS mutation and the other one was KRAS wild type. Just that simple KRAS mutation controlled the amount, the way reovirus infected and the way reovirus propagated and grew within the cells. And these all beautiful structures were the structures which are called the viral arrays. They form a crystal-like structure. So while we were studying, we did notice differences within the cell, but at that point, a scientist also has to look at it from different angles. At that point, we did not really put much emphasis on the structure which I'm showing right now. This was, we understood that this was a consequence of infection and the cells were dying, and we did see many defective mitochondria and endosomal structure, but we did not focus so much over here. We were mostly looking at the viral particles, counting them, and the graph here shows the significant difference in the propagation of reovirus in the two cell types. At that point, the phase 1 clinical trial was also in progress. So we had a patient biopsy. So a typical patient biopsy after reovirus administration showed this type of morphology. This is cryo, so it's not so clear and not so prominent, the subcellular organelles, but we could see some little holes here and there. So I was a little bit discouraged at this point. This patient had received quite a few cycles of reovirus treatment, and I was hoping to see the viral array, and that was my expectation. But on the contrary, what I saw was not too many viral particles and big holes in the tumor cells. So we looked into, now we moved from cryo to regular uranyl acetate sections. And what did we see? We saw similar hole-like structure, and we were seeing that there was a total loss of cytoplasmic integrity, and and the parts of the cell was breaking off. So this is now a typical tumor cell with better contrast. But this was cycle 12 biopsy sample. So this patient had already received 12 cycles of reovirus. But I was trying to look at reovirus, but I as I understand that at that point we were trying to see if the cell lines were replicated here, but these were human samples and they were different. Then when I went up to higher magnification, what I started to see was these little particles. So when I saw this, we realized that this was. We also did a immunogold staining to understand if these were the viral particles. So we started to see more and more of these. So these are the viral factories. This is where the virus grows. And then once we were able to recognize the viral particles, we could see them all throughout within the cells. And there were all these little holes and the viral factories. Either the viral, after the virions matured, they left, making holes on the cells, or some were active viral factories where we could see the virus being made in the process. So then we started to see the biopsy. So now we realize that what we saw in the cell lines which were infected and the biopsy where the reovirus was administered intravenous and the virus had to travel to the cancer cells, their morphology was slightly different. And this is how they harbor in the tumor cells and grow and propagate. So after finding several such structures, we call them viral factories, and we started to define the nature of these factories. They are hollow vacuole-like structure which are membrane bound. They does not seem to contain too much of cytoplasmic component, and they were much lightly stained in by as seen by transmission electron microscopy. So a typical dying cell it's not the cell rupture as is shown by many cartoons, but there is a progressive loss of cellular integrity with discrete holes in the cytoplasm, which consequently leads to dampening of cellular proliferation. We also see the DNA, a lot of heterochromatin, so DNA transcription and other things are reasonably shut down. We see the holes and we see how the cellular integrity is being destroyed. So with this, we move on to autophagy, which is the main topic of our discussion today. So we started from apoptosis. At that point, we did not consider autophagy. Autophagy, we started to test just as to see if any of the autophagy proteins were upregulated. And interestingly, we found out this whole process. So usually what happens, what is autophagy? Autophagy is self-eating, now more and more appreciated that when there is starvation, the cell picks up its wrong proteins and picks up its junk within the cells, processes it, and prepares amino acids and other raw materials for the cell to survive. Autophagy typically is triggered with starvation and fasting, which is an old ancient practice in India, is such a healthy practice. Now more and more people in the West are realizing it, that fasting does help the cells to clean themselves up. So autophagy has a very tight threshold. When the stressor comes, whether it's starvation, whether it's wrong drugs, whether something that the cell does not like, autophagy tries to clean it up. It tries to maintain balance, and it maintains the autophagic threshold. It goes through adaptation. Then it reaches a critical point when it realizes that it is not possible for the autophagy pathway, the autophagy mechanism, to keep the cell alive and healthy, And at that point, if the cell is allowed to survive, it can get transformed. So autophagy then triggers apoptosis, and the cell is killed by programmed cell death, which is the apoptosis. So there is a tight balance between autophagy and apoptosis, and at the apoptotic threshold where the cells try to survive till a point and then it gives up. So when we started to look at, oh, I just really want to quickly go through this slide. So this is where it explains how autophagy happens. So this is endoplasmic reticulum, a protein which is wrongly folded or a protein which has been with a wrong sequence. It's with a mutation in the transcript and a wrong sequence. What happens, as soon as the cell realizes that it's the wrong protein, it tries to chaperone it, it tries to encompass it, tries to protect it from doing something wrong. And there are series of proteins which gets involved in the process. The ones we see are the ATG proteins, ULK1. These are the proteins which helps in initiation. Initiation is followed by nucleation. This is a typical pathway for autophagy. Here we see another important protein, the Beclin-1, the VSP34, and they do the nucleation. At the point of nucleation, the wrong protein is trapped within. And then we see the LC3. LC3 is still considered one of the most important protein involved in autophagy. LC3, changes in LC3, flux of LC3 is considered as a marker, as a readout for autophagy initiation or autophagy inhibition. So here we see with elongation, LC31 is active, and then with several other ATGs and LC32, autophagosome matures. When it completely matures, it's called an autophagosome. This autophagosome then gets fused with lysosome to form autophagolysosome. Autophagolysosome is when it is ready to be processed. The contents of the autophagosome is now being processed. Lysosome, as we all know, is the cleaning organelle for the cell, and lysosome has many acidic proteases and other components which are capable of processing the wrong proteins. So then it's broken down and then it is sent out as the precursors. These are now good ingredients which can go back and help the cell for its survival, making of new proteins and so on. So autophagy, as I said, is a self-eating process, a self-cleaning process. But then when it realizes that when it fuses to form the phagolysosome, and it realizes that this degradation process is not sufficient and it requires much more attention, then instead of doing the recycling, it triggers the apoptosis. When apoptosis is triggered, lysosome ruptures and the programmed cell death process is initiated. So we wanted to study if autophagy is active when reovirus infects the CRC cells, and is there any difference between its action in KRAS mutant versus KRAS wild type cells. So incidentally, at that point, in my lab, things were very active, and many projects were working together. So we did not have any crisis for ROB1. One was the CRC cell lines, HCT116, as I previously mentioned. It's one of my favorite cell lines. It grows very well. It is very aggressive in nature, and it has a KRAS mutation. Along with it, we used HKE3, which is isogenic to HCT116, and it's a KRAS wild type. We used reovirus infection at 5 MOI, and we tried to study autophagy and the expression of autophagy-related proteins at different time points, 3, 6, 9, 24. And we found that at 6 and 9 hours, we saw the most prominent effect. Since cell lines are easily available, and experiments could be repeated quite a few number of times. We did maximum studies with the autophagy, with the cell lines, and we studied the expression of autophagy proteins by immunofluorescence, by flow cytometry, Western blots, as well as qPCR. So this was a very comprehensive study. We also studied it in syngenic mice models. Syngenic mice models are models where the mice are allografted with the CRC cell line. So CT26 and MC38, these are mouse cancer cell lines. The CT26 has KRAS mutation, MC38 has KRAS wild type. Each of these cells are allografted to mice, respective mice. So for CT26, it's BALB-C. For MC38, it's C57 Black 6, the house mouse. And Reovirus was given to the tumor. So the CRC cells were mouse CRC cells were injected to the flanks of the animals, and since they are allografted, there was much less rejection, and tumors were grown on the flank. It has advantages and disadvantages. The advantage being that you can measure the tumor externally. You don't need to do a PET scan and other things to measure the tumors, and the tumors grow on the flank. But on the other hand, they are not exactly the way we want it, that the tumors are growing in the colon within the colonic epithelium. Hence, but syngenic models are quite often studied, and especially in the initial stages like this. So we, at that point, reovirus was given intratumorally. So the tumors were injected with reovirus at a daily dose of 10 million. Samples, the tumor samples were collected at the end of the experiments, at the end point of the protein, and Western blot analysis and qPCR was done. At the same time, in the clinical trial was on, and we had 5 KRAS mutated CRC patients which were receiving reovirus, and we compared their data with 3 KRAS wild type CRC patients. So we had a total of 8 patients divided into 5 and 3, and we wanted to see the statistics. This was a phase 1 trial. Phase 1 trials are usually to look at the toxicity, and an enrollment of about 10 is enough to run the trial. So the trial was on, and the patient and the KRAS mutated CRC patients received a 60 minutes infusion for 5 consecutive days for each cycle. So the cycles were repeated every 28 days, and within that 28 days, for 5 days, they received an hour of infusion of reovirus in PBS, and it was at a dose of 3 into 10 to the power 10 particles, virus particles per day. The PBMC was collected from these samples at different time points at day 2 post reovirus administration, day 8 and day 15, and we did a transcriptome analysis. And what I previously showed you, one of the patients or 2 of the patients gave needle biopsies and core biopsies, and we could do an electron micrograph, and that is where we saw that the previous data which I had shown. So coming to the cell line studies, immunofluorescence microscopy at 6 hours clearly showed that there was expression of LC3 protein. LC3 was FITC labeled LC3 protein, and we had the cells labeled in DAPI. DAPI gives a nuclear staining, so it shows the presence of the cells. And then we had FITC stain, and at 6 hours, HCT treated with reovirus had clear expression of LC3 proteins. This is the overlay of DAPI and FITC, and we can clearly see the puncture or the expression of LC3 protein as an indicator of induction of autophagy. The same thing was not at all when HKE3 was treated with reovirus. No such results were seen. When we counted it and we made a statistical analysis, we found that HCT116 had a 3.4 times fourfold more expression of LC3 upon reovirus administration as compared to the KRAS wild type HKE3. Similar results were also seen at 9 hours. Exactly same thing, more prominent. And here I have the figure so that this is just what you saw at a smaller scale, right? In the previous slide. Here you can clearly see the nuclear staining of the DAPI and the expression of LC3 as green puncta throughout the cells. So this is HCT post-reovirus at 9 hours, and we did not see this in HKE3, but we could very well see it in HCT116. So this was our first indication that uh, reovirus causes the induction of autophagic pathway in CRC cell lines with KRAS mutation. So next we performed flow cytometric analysis. Here also we used LC3, FITC labeled LC3 for the studies, and if you would see the graphs clearly showed that there was more than twofold expression of LC3 as measured by flow cytometry at 6 hours and 9 hours for the HCT116 with KRAS mutation. So we already started getting indications that reovirus induces autophagy in KRAS mutant cancer, CRC, as opposed to the KRAS wild type cancer. So this is at 9 hours, the same results, LC3AB expression. Next, we moved into Western blot analysis. We studied quite a few proteins, LC3, ATG5, P62, VSP34, Beclin-1. These are the proteins which I also showed and marked when I was trying to explain the autophagy process. These are important proteins which are involved in nucleation and maturation of autophagosomes, and we did see a significant fold change increase in HCT in most cases as compared to HKE3. VSP34, we did not find any significance at 6 hours, but we did find significance at 9 hours. So there was a chronology of the different proteins which were induced at different time points post reovirus infection. Now moving to the animal studies, we used 5 animals which were not treated with reovirus and which were treated with reovirus. If you would recall, this is the syngenic model. The CT26 is the CRC cell line, mouse CRC cell line with KRAS mutation. MC38 is with KRAS wild type, and we ran we did a Western blot analysis for LC3AB, and we found that control as compared to reovirus in CT26, as you can see, there was a much higher expression in CT26 when treated with reovirus as compared to MC38. And here is a graphical representation of the densitometric scanning, where we saw a 2% versus 8% intensity or expression of LC3AB in CT26, whereas almost similar expression in MC38. Again, clearly indicating that it's not only the cell lines, but also in syngenic animal models when reovirus is administered, we see the similar effect. Now, reovirus, you might ask, it is not human specific. It has a dual character. It equally infects mouse and human cells. So we were lucky we did not need another strain of the virus, but we could use the same virus for the cell line studies, animal studies, as well as human beings. So here we see the expression of autophagy genes by qPCR at 6 hours and 9 hours for HCT and HKE3. So most of the autophagy related genes that we see here are upregulated in HCT116 were actually downregulated in HKE3. And we also have LC3 over here, MAP1 LC3, that's the LC3 protein, which was upregulated in HCT116 and significantly downregulated in HKE3 at 6 hours. We also saw a difference in proteins. The only protein that we saw was slightly upregulated was ATG5 at 9 hours for HKE3. Otherwise, we saw the same pattern of induction of autophagy proteins both in HCT116 as compared to HKE3. When we looked at the same gene expression, same set of gene expression in the mouse model, CT26 and MC38 syngenic mice model, we found 13 we found 13 proteins, autophagy related proteins, which were significantly overexpressed in CT26 and not at all expressed in actually with a less expression in MC38. And that, of course, includes the LC3B protein, and they were all significant. We had the Richter protein, which is involved in the mTOR pathway. We had the ULK proteins, ATG4 protein. So all these were the important proteins for the autophagy pathway. So by now we were quite confident that autophagy, that reovirus is inducing autophagy in more significantly in KRAS mutated CRC cells. Now we move to the transcriptome analysis. For the patient samples, we did a transcriptome analysis for all the 22,000 genes, and we then selected the 88 autophagy genes, which we studied for the animals, and we wanted to see the consequences and their expression in the 5 patients at days 2, 8, and 15. So these were the times when the PBMC was taken. Here we could not get the biopsy samples at every point because that's not allowed, that's not possible. So we used PBMC as a surrogate for the tumor, and we were trying to follow the induction of autophagy genes in the PBMCs. So what do we see here? We see the data is over here, 48 hours, which is day 2, day 8, and day 15. And we see the expression of these genes in, again, Rector and the LC3B and ULK, all these genes being induced and significant. All these values are significant and were expressed in the patient samples that had KRAS mutation and were receiving reovirus. None of these genes were induced in patients who were KRAS wild type. So we were now confident that autophagy is being induced in actual patient samples, in cell lines, CRC cell lines, and in mouse model. So we did a comparative analysis of the expression of the autophagy genes across the 3 models, the mouse model, the cell lines, and the patient samples. And we found the 3 important autophagy-related genes, the LC3, the Richter, and the RASD1, were commonly and significantly overexpressed or induced upon reovirus administration in the CRC environment. And we established that autophagy is indeed induced. So now we conclude that we had previously shown that apoptosis is being induced. So now we understand that it's initially autophagy that is being induced, and then when the viral propagation continues, then a point is reached where autophagy realizes that it is no longer possible for it to self-eat or to destroy all the viral particles which are growing aggressively. So at that point, it triggers apoptosis, and that causes the oncolysis or the killing of the cells. This propagation does not happen in KRAS wild type cells, hence they are they are not destroyed. So now at this point, we are going back to our transmission electron microscopy, and we are trying to now understand that those figures where the autophagosomes which were formed, they typically match the autophagosome. I did not want to bring it the third time. I showed you the figures twice already, but I mentioned to those special structures, and those were actually the autophagosomes. And the formation of autophagosomes were highly increased in KRAS mutant cells, and when Reovirus was administered. So now we are going back and we are trying to count them and do a quantification between the cell lines and the patient samples. We do not have too many biopsy samples because there were few people who got enrolled and none of them are alive. But whatever sample we have, we are trying to analyze them at this point. So, as a conclusion and for the future direction, we are utilizing the three model systems. Utilizing the three model systems, we have established that reovirus induces autophagy, preferentially in KRAS mutated CRC environment. So, the next step, what is the next step? This finding, how does it help? How does it improve? So, the next step is to study the effect of autophagy inducers like rapamycin, carbamazine, or tadbecklin along with reovirus in KRAS mutated CRC and analyze for survival and tumor regression. So we have already initiated it. The IRB protocols are being in place, but we need to study the effect of autophagy inducers in animal model before we move further. Initially, a few years back, chloroquine was studied in CRC as a inhibitor of autophagy. At that point, there was a hypothesis that autophagy actually accentuates the cancer, so inhibiting autophagy would help in reducing the aggressive growth of tumor cells. But that project, that finding totally failed, and chloroquine failed to have its effect. We now want to see the other way around, that does inducing autophagy, and when using in conjunction with reovirus, does it have the therapeutic potential, and is it more efficacious as compared to other available treatment options? So this analysis then necessitates that we get a robust genetic mouse model that actually recapitulates the human colonic adenoma carcinoma etiology, and can be used in the laboratory as a preclinical study model before we move onto the clinical settings. So we have already achieved that, and I know I'm talking for quite some time now. It's becoming lengthy, so I'll quickly go over it. So we generated a mouse model for KRAS mutated CRC. We use the CRE CDX2 CRE. So CDX2 is a colon specific promoter. So we wanted the KRAS to be expressed only in the colon and not be expressed in the entire animal, because that will have other detrimental consequences and will not allow us to study the specific the consequences of the mutation in the colon. So we used the CDX2 CRE, and we used a KRAS mutation, and we selected APC. APC is the tumor suppressor gene of the colon, which is mutated in 80% of the CRC, and the mutation happens very early on at the time of initiation of CRC. So we wanted to have APC mutated, KRAS mutated model of the animal, which cannot be constitutive, because then it will be embryonically lethal. So it has to be inductive. And we introduce the tamoxifen cassette, ERT2. So with the prior to giving tamoxifen, these animals are absolutely normal like any other mice. But once we give tamoxifen and the CDX2 CRE is inactivated, the mutant proteins are released, and once the mutant proteins are released, the tumors develop, and then we can use these animals to study the platform. So this work was published in 2019. So essentially, in one step, we generated KRAS has to be heterozygous. Homozygous KRAS is lethal. So and KRAS being oncogenic, a single allele mutation is enough to have the detrimental consequences. Whereas APC being a tumor suppressor gene, it has to be both alleles have to be mutated to have the detrimental consequences. So we created the KRAS flox and gene and the APC flox, and we created our KRAS APC animals. We also created the CDX2 ERT2. ERT2 is the cassette which is sensitive to tamoxifen, the Cree with APC flox, and then we back crossed it, and then we had the final model, which we call the KPC APC. This has been characterized, and it absolutely matches. Let me see if I have that slide or not. So it does develop cancer upon tamoxifen administration. We see the HNE over here. So this is only APC mutation. Here we see that we can still see the crypts. We can see the very nice goblet cells. Whereas in KPC APC, which has KRAS mutation, and both the transgenes were activated at the same time with tamoxifen, we see total loss of colonic integrity. Everything is out of place and cancer has initiated. So we have this now. This animal is in holding in Jackson so that they can sell it to other people who are interested in using this model and trying to study different drugs for colorectal cancer. So it has an advantage. It is till date animal model which is most close to the human CRC with KRAS mutation. So with this, I'm going to finish my presentation. This is the last slide, and I just want to. This is my dream, that is to do reverse genetics of Reovirus. While I was writing and making proposals, many of them were turned down. And after the pandemic, I'm sure nobody wants another laboratory-made virus to be out in the society, in the community. So probably I would not be able to do it, or maybe I will try, but doing reverse genetics will help in increasing the oncolytic properties of reovirus. And there are hurdles, of course. It's a double-stranded RNA genome with unknown polarity. We don't know which of the segments which of the strands is the coding strand, and a lot has to be manipulated. And we have a double-stranded transcript. There's no DNA intermediate, so we don't know the template and the coding strands. So these are the difficulties, but I am studying. I have been studying. The crystal structure is solved. The genome details are all available, and it might be difficult, but just changing the genetics a little might make reovirus much more stronger and a much more potent oncolytic agent. We have to wait to see what happens in the future. Finally, the most important slide is the slide of my acknowledgements. Without my collaborators and students, this work would have been impossible. Michael Gerber is the one who did all the molecular simulation with Schrodinger with me during the pandemic. Thong Thai is now doing PhD. He was the technician, but he was helping me in every way to complete the Autophagy project. Elisha did the transcriptome analysis of the patient sample, and so did Avishai. Jishan and Rishika, they performed the qPCR for the animal samples, and that's where I got all the animal data. Collaborators, Dr. Goel I have been working with him for past 10 years. He is the oncologist, and he provides us with the patient sample. He provides the lab with the patient samples and patient data. Dr. Liu is the pathologist who confirms that yes, it is the cancer. Devika was a fellow when the autophagy project was running. Now she's an oncologist, attending oncologist at Sloan Kettering, and Tito Augustine is my colleague in the lab, and we all work together. Also, my thanks goes to the analytical imaging and the micro PET core facility at Albert Einstein College of Medicine. Last but not the least are the patients who enroll in the study and contribute samples for analysis. And finally, I would end with a big thank you to the organizers again of this meeting, and for the time and attention of my viewers. I thank you all, and I'm now open for questions. Thank you.
There aren't any question at the moment in the chat box. If we do find anything, we'll send it over to you. So on behalf of Department of Microbiology, Tripura University, I would like to thank Dr. Moitro for this interesting, informative talk, which has been presented to the students and the entire crowd in such a lucid manner that those who have not really specialized in the area would also be in a position to understand. The way we have organized the talk series is that for each of the talk that we are going to have, at least for the students of Department of Microbiology, the third semester, we are going to have a small questionnaire that would be given, or rather a question that would be placed to them, and within 30 minutes they have to respond back to it. So students, the question for today: what are the new informations that were obtained about the reovirus induced autophagy from the work that has been presented by Dr. Moitro? So your time starts now. It's 12:19 and you have 30 minutes to send back the responses to my email. Other than Dr. Moitro, we are really thankful to you. I would also like to mention and put on record that there are other peoples who are perhaps not on the screen. Now you can't see them, but they have been there on the background, and they include a scholar, Ms. Mandakini Gogoi, a student, Mr. Ajay Modak, and the senior technical assistant, Mr. Shumon Dash, who have been instrumental in the different, works that have to be done for making any of these online interaction successful. And again, before we end, thank you, Dr. Moitro, for all your time, and thanks for always being there whenever we wanted you for our students and the department. Thank you so much.
Thank you, Sham. Thank you.
Thank you. So we'll end here, and the next series, the talk for the next series, would be on first. So see you again then. Thank you, everyone. Have a
Bye.
nice day.